Good afternoon, one and all. Myself, T.S. Almesh Goda. I have completed my undergraduation from University of Horticulture Science, Nagalpur, and I have completed my MSc from the Division of Food Science and Post Harvest Technology, IIT New Delhi. First of all, I'd like to welcome you all the participants for All India Horticulture Entrance Exams Coaching Series. I will be taking two classes regarding Post Harvest Technology, and the contents which I am going to discuss is regarding maturity indices, physiology and ripening, enzymes, post-harvest novel treatments, and also morphological classification. Before I start post-harvest technology part, one thing I would like to say is, since for a JRF, since there is 120 questions, maximum we can expect 10 questions, 10 questions from the post-harvest part. So since these 10 questions will be very direct questions and to the point question, it will be very easy. I would like to say, if you put 75 to 80% effort for fruits and vegetable and floriculture part, at least if you put 10 or 15% for post-harvest and PMA parts, you can attend all the questions, 10 from PMA and 10 from post-harvest. So this will be very easy question. I would like to say, at least you try to revise once or twice. It's more than enough. And coming to my classes, first is regarding maturity indices. Maturity, hello sir, sir. Hello. Maturity may be defined as its attainment of a proper size after which ripening takes place, whereas ripening means the qualitative changes in the fruits after maturity as a result of which it becomes edible. In maturity, there are two types: horticulture maturity and physiological maturity. Coming to horticulture maturity, it is a stage of development when a plant or plant parts possesses the prerequisites for utilization by the consumer for a particular purpose. Whereas physiological maturity, it is a stage of development when the produce completes the development process by it may or may not ripe. Coming to various fruits and vegetable crops for their maturity indices, coming to specific gravity, Alphonso mango 1 to 1.02, Dashairi mango less than 1, potato tuber for vegetable purpose, it should be 0.98 to 1.02. For chips making potatoes, the specific gravity should be 1.08. For pineapple, 0.98 to 1.02, and guava 0.95 to 0.96. See, for JRF aspirants, they'll ask just as the maturity increases, what happens to specific gravity? Option A, either it increases, decreases, or other two options. Actually, as the maturity increases, the specific gravity of the commodity increases. This is the correct answer. And coming to solidity, it is a cabbage, Brussels sprouts, leaf vegetables, etc. And in TSS part, you might expect one question or a matchup questions, like matching commodity with TSS. In this, the most important is they'll ask kiwi fruit, that is 6.25 degree Brix or 6.5 degree Brix. And in case of resins, it is a most important question is for grapes which we want to make resins. The TSS should be 20 to 24 degree Brix. It should be colorless and seedless variety. And coming to apple, this is also very important question, like apple for cider preparation, the TSS for that particular apple variety should be 14 to 17 degree Brix. And also bale, it should be 30 degree Brix. And coming to lychee, it is a flattening of tubercles, pineapple flattening of eyes, banana disappearance of angularity, juiciness sweet corn and citrus 35 to 50%. Netting, muskmelon is a major maturity indices in muskmelon. And from days from fruit set to harvest, it's pomegranate 135 to 170 days, banana 90 days, alfonso and pyri mangoes, it's 110 to 125 days, sapota 300 days. In apple, they'll ask the most promising maturity indices for apple. If they ask, if they give the most of the chances, the correct answer is days from DFFP, days from full bloom to harvest. It's 125 plus or minus 4 days. And in mango, tapka stage, this is also very easy and direct question in JRF, they'll ask tapka stage of maturity is related to which fruit crop? It's mango. Coming to sugar to acid ratio, this actually yesterday when ma'am was teaching, some of the participants asked that ma'am regarding TSS to acid ratio for various citrus fruits. It's for sweet orange, it's 8.5 to 8.9, mandarin 10.5 to 13.0, and quinoa mandarin it's 12.1 to 14.1. Quinoa mandarin, this question was asked in JRF 2019. When we wrote the JRF exam, they asked the most promising maturity indices for quinoa mandarin is TSS to acid ratio, TSS acid, and other option. The correct answer is TSS to acid ratio. And next is pulp to peel ratio of the banana. It should be 10.1. And for lychee and pomegranate, Briggs to acid ratio should be 70 is to 1. And the starch iodine test, it is the most used in apple for detecting the maturity indices of apple. For delicious group, it should be 4.5 by 10. And compactness, cauliflower head, brussels sprouts and broccoli. And okra, it's also a question they'll ask for harvesting, the okra length should be 6 to 7 centimeters is the correct answer. And calyx drying is one of the most prominent maturity indices for Cape gooseberry fruits. And also coming to others, here I did not include like change in the color. This is a common in all fruits, so I have included only the important one. And also coming to walnut, package tissue brown stage. It is also one of the important maturity indices. Coming to flower crops, when you see rose, it's 1 to 2 petals begins to unfold at the tight bud stage. Just matured, unopened bud stage. Anthurium, see, this is also important question. They'll ask 1/3 of the flowers on the spadix should be fully developed and matured. This is also very important question. And coming to anthurium, it's 1/3 florets open. Cattleya species of orchids, it's 4 to 5 days after opening. And chrysanthemum, it should be fully expanded. Coming to dahlia, it is also fully expanded flowers. Coming to carnation, the paint brush stage is the most promising stage for harvesting carnation. Coming to gerbera, the flower should show the petals should shed the pollens. It should be fully mature and it show the pollens. That is a major in a gerbera. And coming to bird of paradise, the first flower should be open. China aster, it should be fully developed with the long stalk. And in tuberose, when few flowers open at the base in the stalk, when few flowers open at the base, it's a maturity indices for harvesting of tuberose. Coming to tuberose of open colored bulbs, liliums, it should be colored buds, and cymbidium orchids, it should be 3 to 4 days after opening of all the flowers. And coming to next part is physiology of ripening and maturation ripening. As we know that as a ripening of the commodity, either it might be a fruit or vegetable, as it starts to mature, the chlorophyll degradation takes place with the help of chlorophyllase enzymes. Then various pigments develop, various aroma in a commodity, various acidity of the commodity as on the ripening, acidity of the commodity decreases, except in banana and pineapple, acidity increases upon the ripening. First, when we come to major pigments, cyanidin 3-glucoside, it is major in pomegranate, plum and blackberry. Cyanidin 3-rutinoside, cyanidin 3-glucoside, it is major in sweet cherry. Pelargonidin 3-glucoside in strawberry. Malvidin 3-glucoside, blueberry. Pionidin 3-glucoside, cyanidin 3-glucoside, malvidin 3-glucoside in grapes. Delphinidin 3,5-diglucoside and petronidin 3,5-diglucoside, it is jamun. From this part, they might ask one question that which pigment is present in which fruit crop. Okay. Then coming to carotenoids, lycopene is majorly present in tomato, watermelon, guava, pomelo, cultivars like star ruby and deep red. Coming to beta carotene, it is major in peach, nectarines, apricot, loquat, Cape gooseberry and also pumpkin. It's a major pumpkin and carrot also. Capxanthin in red pepper, violozanthin in orange, caricazanthin papaya. It is the most commonly asked question that caricazanthin is majorly found in which fruit crop? It's papaya. Then xanthin, it's in mandarin, lemon, Rangpur lime. And also with changes with respect to aroma and phenolic compounds. First, ripe apple. Ripe apple aroma is due to ethyl to methyl butyrate. Pineapple, it's ethyl butyrate. Ripe banana and green banana, it's different. For ripe banana, it's eugenol, and for green banana, it's hexanol. And for pear, it's butyl butyrate. For apricot, it's hexyl acetate. For lemon, it's citral. For grapefruit, notaketone. It's also most commonly asked question that the flavor or the aroma of the grapefruit is due to dash. It's notaketone. And orange, it's valencin. Guava, beta carotene and beta alanine. And these two are very important with respect to grape is that muscat flavor of grape is due to, they'll ask like muscat flavor of the grape is due to dash. It's due to methyl salicylate, and foxy flavor of the grape is due to methyl anthraquinone. These two are most important question, like which is the muscat and which is foxy flavor of grapes. Coming to phenolic compounds, the yellow and brown color of see, while we study we'll get more confusion like color regarding the color of the onion, like yellow and brown color of the onions, which is a major pigment, major phenolic compounds, or red onions, which is a major pigment. So this is a correct this one phenolic compounds. First is yellow and brown onions, it's quercetin, and soya genistein is majorly present in soybean, and anthocyanin. See, this is also most asked question, like anthocyanin. The red color of the anthocyanin shows in which pH, like acidic pH or basic pH. They'll ask like this question. The correct answer is acidic pH. At acidic pH less than 6.5, the anthocyanin shows a red color. More than normal pH, a neutral pH, more than 7.5 or 8 pH, the anthocyanin color from red color it changes to blue or brown color. So majorly grape, pink and red onions and jamun are majorly is due to anthocyanin, and cyanidin is majorly found in red cabbage. Delphinidin is majorly found in rose. We can see from genetic engineering, this color has been taken from the petunia flowers for rose delphinidin color. Coming to malvinidin, it is majorly found in blueberries. Ellagic acid, it's majorly found in pomegranate, mustard and strawberries. Vanillin, it's majorly found in vanilla. Lycopene, see, they'll ask red color of the carrot is due to dash. We should not take like carotene. We should not get confused. The correct answer is lycopene. Red color of the carrot is due to lycopene and also tomato. And resveratrol is a major compound present in red grapes. And coming to see, as the maturity or ripening takes place, there will be a change with respect to organic acid and also sugar. Sugar content increases and organic acids decreases. The major is in grapes, the major sugar is glucose and organic acid present in tartaric acid. In strawberry, blueberry and persimmon, major is fructose and citric acid. Here in some of the paper phenol you will get the question like which is the most sweetest sugar. It's fructose sugar followed by sucrose followed by glucose. Okay. Next is peach, nectarines, plum, apricot and loquat. Here the major sugar is sucrose followed by organic acid is malic acid. In pomegranate and sweet cherry, major sugar is fructose and organic acid is malic acid. They'll also ask like major acid which is responsible for respiration is, it's a malic acid. They'll give like which is among options like tartaric acid, citric, malic acid. For majorly respiration, malic acid is more suitable. Coming to all is only in this like blueberry, raspberry, jalebi, kiwi fruit and apricot. In organic fruits, cherry, lychee, nuts, loquat, carambola, olive and onla. Only the confusing one I have included in climatic and non-climatic section. Based on the fruits according to respiration rate. As you all know, as the maturity takes place, there will be higher in respiration rates. Coming to respiration rates, here they'll ask like in apple. Apple, based on the classification on respiration rates, apple belongs to which group? It is the most question. Apple belongs to low category. And coming to banana and mango, these two are also most commonly asked question, like banana and mango. When compared to respiration rate and ethylene evolution rate, banana and mango always belongs to moderate. You keep this in mind, like banana and mango always belongs to moderate rate in both respiration rate and ethylene evolution rates. But in apple, when we come to respiration rates, apple it belongs to low category, whereas in ethylene evolution rates it belongs to high category. I'll come to next that. First one is when come to low category, very low category, it's nuts, dates, dried fruits and vegetables. Coming to low, it's apple, citrus, grapes, garlic, onion, potato and sweet potato. In moderate section, majorly apricots, banana, cherry, peach, pear, nectarines, fig, mango, cabbage, carrot, lettuce, pepper and tomato. Coming to high category, it's strawberry, blackberry, raspberry, cauliflower, lima bean, avocado. In very high category, it's globe artichoke, snap bean, green onions, Brussels sprouts. In extremely high, these also they'll ask extremely high it belongs to majorly mushrooms, broccoli, asparagus, pea, spinach, and sweet corn. evolution rate. Majorly there are five category like very low, low, moderate, high, and very high. Coming to very low category, majorly globe artichoke, asparagus, cauliflower, cherry, strawberry, pomegranate, leafy vegetables, and potato. Okay. Coming to low category, it's blueberry, cranberry, cucumber, brinjal, okra, olive, pepper, persimmon, pineapple, pumpkin, watermelon. Coming to moderate category, majorly banana and mango. These are important questions which they'll ask in exam. In I category, as I said, apple belongs to I category and kiwi fruits. They'll ask apple belongs to which category in you should read the question correctly like apple belongs to which category in ethylene evolution rates and respiration rates. Okay. Coming to high category, it's cherimoya, passion fruits, and sapota. And these also very important question like they'll ask adverse effect of ethylene on different crops. Like lettuce, you can see an adverse effect of ethylene. There will be more research spotting, discoloration. In carrot and beetroot, due to formation of isocomerins, there will be development of bitterness. In celery, it affects blanching. In cabbage, there will be leaf abscission and development of undesirable flavor. In asparagus, it will be fiberness. In cucumber, degreening is a major problem due to adverse effect of ethylene. In Brussels sprouts, there will be stem elongation, and in peas there will be development of toxin pyzatin formation. Okay. In mushroom there will be stalk cap development. See, when the stalk and cap opens, there will be then the commodity loses its shelf life very fastly. So it is also major adverse effect of ethylene. Coming to sweet potato, there will be hardcore and production of phenolics. And in potato, there will be suppression of sprouting. In tulips, bluing of petal. It is also most commonly asked question like bluing of petals in tulip is mainly, the major factors for bluing of petals in tulip is mainly due to adverse effect of ethylene. In strawberry, there will be increased rotting. In tomato, there will be increased lycopene and carotene development. In carnation, sleepiness. It is also important question there as sleepiness in carnation is due to ethylene. Okay. In second box, it's very important like ethylene forming enzyme. ACO enzyme is a ethylene forming enzyme. Ethylene biosynthesis inhibitors are ACC synthase, ACC oxidase, polyamines like putrescine and spermidine, and ethylene action inhibitor like 1-MCP, 1-methyl cyclopropane, and silver thiosulfates. These two are the major ethylene action inhibitors. They'll ask like which is the most commonly used ethylene action inhibitor in cut flowers. STS, that is silver thiosulfate, is a major ethylene action inhibitor in cut flowers. It is one of the important question. And coming to precursor of ethylene, it's methionine. So you should get to know that which are the precursor of oxygen, which are the precursor of gibberellin, precursor of phytokinine, ethylene. You make one column and you just go through it because it's also a very important question. You can expect in a matchup following, like which are the precursor of which compound. It is also a very important question. And coming to enzymes in the food industry, these are not much important to JRF aspirants, but for SRF and ARF, I think it is very important because they'll ask enzyme and which is responsible for which factor, why it is used. Coming to amylase enzyme, it's majorly used in bread making, pre-cooked baby foods, breakfast food, and for making mashing and also syruping. Coming to cellulase enzymes, it's majorly used in almost all the food commodities. Coming to glucose isomerase, it's majorly used in sugar and starch compounds, and also glucose oxidase. It's used for removal of glucose and oxygen. And coming to invertase, it is for SRF aspirants in post-harvest technology. The most commonly asked question is the soft centered candies. The major enzyme present in soft centered candies is invertase. It is one of the important question. And also in lactases, lactase is present in milk. It is present in almost all the bakery items like milk concentrates, ice creams, frozen desserts, etc. And in pectin, they'll ask when it comes to JRF, they'll ask like clarification of the juice is mainly done by which enzymes? It is pectinases enzymes. Okay. And coming to coffee beans, it's like they'll ask one common question, frequently asked question is the flavor or the aroma of the coffee beans is mainly due to dash. Like the correct answer is roasting. Making of the roasting makes the coffee beans to develop a very good aroma. It is also a most commonly asked question. Coming to renin enzyme, it is majorly used in cheese production. Coming to protease enzyme, it is majorly used in bread making and also meat tenderizing. In meat tenderizing, they'll ask like papaya. You can see in papaya there is a papain enzyme. It is also majorly used in meat tenderizing. Okay. And coming to post treatments, that is chlorinated water. See, when you harvest the commodity, mainly tuber crop, the washing of the commodity is very must in order to avoid the damage by fungi or the bacterial enzymes. So, washing with chlorinated water is very required, is much required. The concentration of chlorinated water is 100 to 150 ppm. It's also very important question. And if you use more chlorine water, there will be a development of toxin like trihalomethane. Okay. In when we come to chlorinated water, they'll ask only three questions. That is, what is the concentration of chlorinated water? It's 100 to 150 ppm. What should be the pH of the chlorinated water? It is neutral pH, like 6.5 to 7.5 pH. And if you use a higher amount of chlorine, what is the toxin developed is trihalomethane. These three are from chlorine. And also coming to oxygen, it's either used in the form of oxygen gas or ozonated water. Okay. Coming to irradiation, the irradiation is the most frequently asked question. It is a very important irradiation. I'll come to this in next slide. And in hot water treatment, see, some of us will get confused like what is the temperature required for hot water treatment and what is the temperature and the time required for hot water treatment. It is 45 to 55 degrees Celsius for 5 to 60 minutes. Okay. In vapor heat treatment, it is also very, will get in one book, there will be different temperatures and different times. But the correct answer is, it's 50 degrees Celsius for 20 minutes. The vapor treatment is 50 degrees Celsius for 20 minutes, but the pulp temperature, it should reach 47.5 degrees Celsius. Okay. And bioagents you can see, we are going to use in order to control various microorganisms. The main action behind the bioagents is, you might see antibiosis or anti-zoonosis factors, various to kill various microorganisms. Okay. And coming to vaccine or coating, in this part, what they will ask is, vaccine and coating of the commodity decreases helps to decrease the transpiration and respiration. It is a direct question. They will ask, vaccine or coating of the fruits and vegetable helps to reduce both transpiration and respiration. Okay. Coming to degreening, in degreening they will ask like for which and all the commodity they might give among the following degreening is followed in which commodity. It is, you remember as BMTC, banana, mango, tomato and citrus. Okay. Best degreening temperature is 27 degrees Celsius, 85 to 90% of relative humidity, and ethylene concentration that is used for degreening of commodity is 20 ppm, but most commonly 5 to 10 ppm is used, and carbon dioxide level should not exceed 1%. Okay. Coming to irradiation, in 2019 JRF they have asked source of irradiation. It's cobalt C60 and cesium 137. Okay. Source for irradiation is important. And coming to this part, in 2020 SRF they asked the blue box, they has same column they asked for Mathifolum because it's value from standard book and it's very important because these readings you should keep in mind. What is the range of irradiation for sprout inhibition, what is the range of irradiation for ripening inhibitor, like that. So these two boxes are very important. Coming to first one, in case of apple, in order to control scald and brown core, 1.5 kg irradiation is required. In case of apricot and peaches, to inhibit brown rods, 2 kg radiation is important. In banana, for inhibiting the ripening, 0.3 to 0.35 kg is required. And in mushroom, to inhibit the stem growth and cap opening, 2 kg is required. In strawberry, to inhibit the gray mold like Potatis cinerea. It is also one of the important questions. They'll ask like what is the range of irradiation that is used to inhibit the gray mold in strawberry. Okay, it's 2 kg. In papaya, for disinfection against the fruit fly, it's 0.25 kg. In case of tomato, to inhibit the alternaria rot, it is 3 kg. Okay, and in this blue box is very important for SRF aspirants candidates who are preparing majorly. It might be fruit peoples are also post-harvest people. It is this box is very important. Like they'll ask this sprouting inhibitor, what is the range? For sprouting inhibitor, it is 0.03 to 0.15. For ripening inhibitor, it is 0.25 to 0.75 kilogram. For insect disinfestation, it is 0.25 to 1 kilogram. Okay, and in case of spoilage against the microorganisms, it is 1.5 to 3 kilogram. In case of elimination of microbes in fresh and frozen meat, it is 3 to 7 kilogram. In case of removal of microorganisms from the spices, it is 10 kilogram. And in case of sterilization of the meat products and hospital sanitization, it is 25 to 70 kilogram. Into this one irradiation dose, this is very important. See, for controlling the mango stone weevil, 300 gray irradiation is required. For preventing mango stone weevil adult emerging from the fruit, irradiation used is 150 gray. For lenticel spotting and surface discoloration of the mango, 600 gray is required. For controlling commercial control of fruit fly in papaya, it's 250 gray is required. For delaying ripening of mango and banana, it's 25 to 50 kilo radiation is required. For inhibiting sprouting in onion and potato, it is majorly common asked question like inhibiting sprouting in onion and potato. What is the range of irradiation used? It is 5 to 10 kilo radiation. Okay. While you see other countries like Taiwan, Japan, and European Union, irradiation is not at all permitted. Okay. When we come to controlling of the potato tuber moth, irradiation used is 20 kilogram, 20 kilo radiation. In effective control of rot in ginger, 15 kilo radiation is used. To control the anthracnose in mango, 105 kilogram is used. Okay, these are very important with respect to irradiation. So I'd like to say at least you try to revise once or twice, because these same values will be asked in exam. If you revise once or twice, you can attend exam. You can attend this question absolutely correctly. Okay. And one thing I should say is in this, when you study the fruit, vegetable, or any horticulture commodity, first try to remember all the major pest scientific name, major pest. For example, if you come in mango, what is the scientific name of mango stone weevil? What is the scientific name of mango thrips? If you come to, for example, if you come to black pepper, what is the scientific name of pollu beetles? You should remember all the scientific names of major pest and also major disease, because it is also they'll ask in exam. For in case of SRF 2020, they asked what is the scientific name of pollu beetle of black pepper, that is Lanka Ramakrishna. So I would like to say you should remember all the scientific names of major pests and major diseases in commercial crops, not in all the crops, in commercial crops. Okay. Coming to vapor treatment, it was first used to kill majorly two fruit flies, like Mediterranean fruit fly and Mexican fruit fly. That is Mediterranean fruit fly is Keratitis capitata, and Mexican fruit fly it is Anastrepha ludens. Okay. Here the mode of transfer of heat, vapor treatment mode of transfer of heat is through conduction. In also hydrocooling, it is the mode of transfer of heat through conduction mechanism. Okay. Vapor heat treatment is mandatory for shipment of mango to Japan, South Korea, Taiwan, and Philippines. It is also most important question. They will ask like, if you want to export mango from India to other countries, for these four countries, vapor treatment is very must mandatory for exporting mango to these four countries. Okay. Coming to hot water treatment, it was first reported by Fawcett in 1922 to control decay of citrus fruits, but later it was used to control insect disinfestation. Okay. Hot water treatment is an approved quarantine treatment for export of many fruits and vegetables against pest. Treatment with 49 degrees Celsius, 49 to 50 degrees Celsius hot water treatment for 20 minutes is most suitable for papaya, lychee, caramel, and also mango. This was approved by USFDA, USDA office from Hawaii. Okay. And coming to use of bioagents to control the post-harvest diseases. In ERS and also in this one SRF, they asked like same question, that Aspire bioagent which organism? It is Candida oleophila. It is used for controlling gray and blue mold of citrus. It is also very important question. And also Biosave. Biosave contains Pseudomonas syringae for controlling blue mold rot of citrus. These two are very important formulations for exams. They'll ask. For JRF, I don't know whether they'll ask or not, but for SRF or ARS or other exams, it is very important question they'll ask. And coming to use of first bioagent, see, most of the members get confused like what is the first bioagent, which was the first bioagent was used to control which disease. Okay, the correct answer is first use of bioagent was to control Botrytis rot of strawberry, that is Botrytis cinerea, by using Trichoderma species. You note it down. This is very important question. First bioagent use was Botrytis rot of strawberry by using Trichoderma species. Okay, the term biological control was first reported by Harry Smith in 1919 for biological control of insects. Okay. And here, when you come to various bioagents to control major diseases of the fruit or a vegetable crops is coming to bacterial section. It's Bacillus subtilis. It is used to control green mold rot, stem end rot, sour rot, brown rot, gray mold rot of citrus, peach, plum, apricot, and strawberry. Coming to Pseudomonas syringae. To control blue and gray mold rot, Pseudomonas syringae is used to control blue and gray mold rot in apple and citrus. In Pseudomonas cepaceae, it is used to control blue and gray mold and brown rot in apple, pear, peach, and nectarines. Coming to Pseudomonas putida, it is majorly used to control soft rot of potato. Okay. Coming to fungi section, Trichoderma argianum, it is majorly used to control blue mold rot, gray mold anthracnose in case of citrus, grapes, kiwi fruit, pear, and strawberry. Trichoderma viridae, it is majorly used to control green mold rot, stem end rot, gray mold rot in citrus, mango, and strawberry. Okay. Coming to yeast section, they'll ask like this formulation belongs to which, like Pichia guillermondi, either it is a bacterial strain or fungal strain or yeast strain. It is also important question. Pichia guillermondi and Candida oleophila. These two are the yeast strains. Okay. These two are very important. These two are important questions. And coming to vaccine question, as I discussed regarding the slide of novel bioagents, novel treatments used in post-harvest vaccine, I told vaccine is used to control both respiration and respiration. That is the only one major question that is asked in vaccine part. Okay. Other than that, vaccine prevents 30 to 50% loss of water. It is used to control transpiration, respiration. Okay. There are majorly two types of vaccines like O and W type. O type, it gives a glossiness to the commodity, and W type, it doesn't give much glossiness to the commodity. And majorly on an average, the wax contains 12% of TSS. Okay. In wax, there are mainly so many types, but majorly there are mainly three types like paraffin wax, Caronova wax, and shellac wax. Okay. The paraffin wax, it is used to control of water loss. It controls a good water loss, but it's having the poor luster. Okay. Caronova wax, it has a good luster but poor control of water loss. Shellac, you can see the best shining property in the shellac waxes. These shellac waxes are majorly used in packing of dried fruits and vegetables like dates, resins, dried commodity. These are majorly packed by shellac wax. Okay. And majorly in coming to our fruit section, they will ask like what is the commercial formulation of wax that is used in mango. It's 4% wax is used commonly. In apple, it is 3% wax is used in apple. Okay. Coming to marketing part, there are mainly two like storage wax and packet wax. Storage wax for immediate marketing, not for immediate marketing, means for storing purpose, storage wax is sprayed. For packet wax, for immediate marketing, packet wax is used. Okay. And in some objective, I read like Ducolitar belongs to which, like it is a wax. I also don't know much regarding this, but Ducolitar is a type of wax. You remember just this point. Okay. Coming to curing part, it is favored by the high temperature and high humidity. Except onion and garlic, there will be 70 to 75% of relative humidity for curing of onion and garlic. Okay. In case of onion, the field curing of onion is called, they'll ask like what is field curing of onion is called as windrowing. The temperature range should be 37.8 degrees Celsius for 3 to 5 days. Okay. For artificial curing of onion, it should be 40 degrees Celsius for 16 hours. For potato, it should be 18 to 20 degrees Celsius at 85% of relative humidity. For sweet potato, it should range from 29 to 33 degrees Celsius with 85 to 90% of relative humidity for sweet potato. And in case of cassava, the curing temperature should be 18 to 40 degrees Celsius. It is also very important. They'll ask anyone, like they might ask onion or potato or sweet potato or cassava. It is also important. And coming to recent updates with respect to post-harvest technology, FSSA has asked to ban the blending of mustard oil, because from previous years we was using the mustard oil, blended mustard oil with sunflower or other oils. So now FSSA has said that no, that mustard oil should not be blended with any oil. It should be marketed as a fine only mustard oil, fine mustard oil. Okay. And coming to this one, I think this we might expect in general agriculture part question or in the posters part question, that trans fat by 2022, from the next year, from the January, the trans fats in the food commodity, it will be reduced to 2%. It should be reduced to 2%, because up to 2021, FSSAI told that trans fats in the food commodity, it should maximum limit is only 3%. But from the next year, they are told no, from 3%, it should be reduced to 2%. Okay, it is very important question. From 3, they are reduced to 2%, because these trans fats are highly health hazardous. These are cancer causing and also neurotoxic. Okay. And another one is FSSA gives more time for the compliance with calcium and magnesium limits in the packet drinking water. And coming to other is for importing. If you want to import the apples from the Washington, so it is must there should be a genetic modification free certificate. Okay, it is also a very important question. Import of Washington mangoes on hold pending genetic modification free certification. Okay, and coming to other, Supreme Court has ordered that there should be a health start rating for food items, majorly beverages. Okay. And coming to another one is FSSAI makes mandatory for food business to mention the FSSAI license number or bills from October 1st from this year 2021. Okay. And there was also honey violence, Indian honey violence. They might have Indian honey violence, whether it took place or yes, Indian honey violence has taken place recently. Okay. And coming to other, the recent bill is FSSAI revised the draft Food Safety and Standards Amendment Regulation Act 2020. Okay. And coming to here, there was held in April 21st, 2021, there was a session held in Codex Alimentarius Commission, Italy, regarding fifth session on majorly spice and culinary herbs. Okay. You just go through this. And coming to other is FSSAI has made Bureau of Indian Standard Certification mandatory for all the packed drinking water companies. Okay. And regarding blended mustard oil, I have told. And one more important is they have changed the logo of when you see the food commodity, you can see the two mark. Either it may be green or red. Green indicates the veg commodity. Red or brown, it indicates non-veg commodity. But what FSSAI has done recent is they have changed the round color mark of non-veg red commodity, non-veg commodity to the triangle, because it is to help the blind peoples. See, by touching, the blind peoples can recognize that yes, this is a non-veg commodity. So they have changed from round to triangle shape of non-veg commodity of red color. Okay. And coming to recently, there was release of GM rubber. In GM rubber, one thing you should remember is majorly manganese containing superoxide dismutase enzyme is used in case of GM rubber. This is important question they might ask. And recently, Bayer's company has introduced a new yellow watermelon variety, that is Yellow Gold 48. It is a yellow flushed watermelon variety. It is also recently released. And coming to IRI and IHR convocation, in IHR convocation, the institute has been released two major varieties, that is Arca apexa in tomato and Arca shuba in marigold, which is a carotene rich variety. 2.8% high carotene content marigold variety is Arca shuba. In tomato, it is Arca apexa. Okay. In IRI convocation, they have released two variety like Pusa alpana, which belongs to rose. It is a pink and fragrant variety. Pusa sundari in chrysanthemum. Thank you. Oh, thank you all.
Okay, thank you, Hanmesh. It was a nice presentation. Students, if you have any queries, you can ask in question and answer, or you unmute yourself and you can ask.
Sir, you potato, the sprouting is due to.
Yes. In potato,
Sir,
hello.
the sprouting is due to.
In potato, majorly sprouting takes place mainly due to variation in the relative humidity and temperature. I think so. Means majorly curing is done to reduce the sprouting and also spraying of malic hydrazide and CFA, CAPC chemical. It is used. Sprouting, I think, is majorly due to relative variation in the relative humidity, temperature, and also irrigation. Before harvesting, before 3 to 4 weeks, you should withhold the irrigation. In onion and potato, in all the onion, potato, or cassava, you should withhold the irrigation 2 to 3 weeks before the harvest. If you irrigate more irrigation, it might also be the reason for sprouting of potato or onion or sweet potato.
Okay, sir.
Sir,
Hello.
Okay, sir.
Hanmesh,
Okay.
In high relative humidity, it favors ripening or delays ripening, sir.
No, in high relative humidity, it will delay means see if you take refrigeration storage or cold storage, if you store the commodity, the temperature will be less and relative humidity is high. We are storing the commodity in the refrigeration in order to extend the shelf life. So automatically, when there is a higher relative humidity, the ripening process will be very slower, very slow, not fully stop. The ripening or other factors which are favoring for the ripening of the commodity, it will be very lesser, so that we can store the commodity for a long period of storage.
Sir,
Any
tapka
other?
stage means what, sir? What's the tapka stage in mango?
Okay, tapka stage means falling of the ripened fruit from the tree. Means when the fruit ripen in the tree, the fallen ripe. See, if you take a one single tree in mango, if there is a falling of 5 to 6 ripened fruits, you think that yes, tree mango fruit has come to the ripening stage. Means it is just the falling of ripe fruits from the tree, tapka stage.
Okay,
Yes, almost it has reached the ripening stage, 80%, 90% ripening. Any other questions?
Sir, you have told that DMTC in a de-greening occurs. What is DMTC?
What?
Sir,
I did not get it.
sir,
Okay.
sir, you told now in DMTC there is de-greening. Then what is DMTC?
No, not DMTC. I told us BMTC means banana, mango, tomato, and it is citrus. Yeah, just as a code word, I told you to remember. Like they will ask in question in exam, like they'll ask in which commodities majorly degreening is done. So for easy remembering, I told majorly in banana, mango, tomato, and citrus, degreening is majorly done. Okay, just as a code word, I told you to remember.
Okay, sir, thank you.
Okay.
So can you
So what
please
is-
repeat the point that you said about the GM rubber?
Yes. GM regarding GM rubber, now wait, I'll open that slide. See, you can see GM rubber. See, GM rubber means it was actually developed by Kerala-based Rubber Research Institute of India. Okay, they have planted this rubber in Assam. They have tested for various multi-location trial, and they have planted in Assam for to see the effect of. Majorly, they have used manganese containing superoxide dimutase. See, you can see the last point. See, it is expected to tide over the severe cold conditions during winter, a major factor affecting the growth of young rubber plants in the region. Means, in order to cope up with the cold environment or cold conditions, these manganese containing superoxide dimutase enzyme is used in GM rubber. Okay.
Sir, what does mean by harvestable maturity and physiological maturity, sir? What's the difference among them?
Okay. See, horticulture maturity is, see, with example, I'll say. For example, if you want to harvest the okra, okay. See, commonly, when you delay the harvesting in okra, it develops a fiberness, right? So, in order to meet the consumer demand, you are going to harvest the commodity earlier, before the ripening stage or before the full development stage. You are going to harvest the commodity as per the consumer preference. That is called as horticulture commodity. Okay. Whereas physiological maturity, it is the stage at which the particular commodity has finished all the cell growth, cell development, and maturation. Okay. Yeah. Say for example, in horticulture commodity, majorly horticulture crops will come. In physiological maturity, majorly cereals and pulses that will come under the physiological maturity section. Okay. Based on the consumer preference, if you are harvesting the commodity, that is horticulture commodity. Like you'll say okra, it should have 6 to 8 centimeter length, or we are going to harvest the okra after 6 to 7 days. I am not sure about the date. 6 to 7 days after the anthesis, so that it will meet the consumer demand. Okay, based on the consumer demand, if you harvest, it is horticulture. After the complete full development stage, if you are the harvest the commodity, it comes to a physiological maturity. Okay.
Okay, sir. Thank you.
Okay. Any other questions or any other doubt? If I am not clear with any other concept, I can say once again. If I am speed or if I am not clear with any concept, you can say so that I can repeat once again.
Sir, while maturing, specific gravity increases or increases only, no? You said.
Yes, specific gravity increases. Yes,
Okay, okay, okay.
specific gravity increases.
So, what would be the best parameter for judging the post-harvest maturity for various crops, sir?
Various crops. Actually, I have told in my first slide regarding maturity indices. No? Like various commodity, like there will be in Kinomandin. I'll just say the most important questions they'll ask in exam. Okay. For when you come to citrus groups like Kinomandin, TSS to acid ratio is a major maturity indices. When you come to walnut, it is packaged tissue brown stage. When you come to Cape gooseberry, it is drying of calyx. Then when you come to grapes for resin making, it is TSS like 20 to 24 degree Brix. And when you come to apple, there are two, that is DFFP, days from full bloom to harvest, and also starch index is also used in apple. And in mango, majorly specific gravity only they will use like this. I have told in first slide of my PPT. The PPT will be shared. Means I just told you the major which they are going to ask in exam. Okay.
Yes, sir.
Sir, the sweetest sugar, sir.
Sweetest sugar is fructose, followed by sucrose, followed by glucose. Okay.
Okay,
First
okay.
is fructose, then sucrose, then glucose.
Thank you, sir.
Okay. Any other questions? If I am not clear, I can repeat once again. If I am clear, then no problem.
Hello, sir.
Yes, yes.
So what is T stage and something about penetrometer like description? What is penetrometer or use?
Okay. T, you are asking regarding T stage, na? Hello. T stage. Okay. T stage, na? T stage, it is a maturity index is majorly used in apple. See, when you see the commodity, whether it is, when you want to see whether the commodity is ripened or not, or whether it has reached the maturity stage or not, if in apple you can see the pedicel, the stalk that is attached to the fruit. Okay. If it is at 90 degree, if the pedicel is straight, and if it is a 90 degree, if the pedicel and the fruit surface, if it is exactly perpendicular, if it is a 90 degree, then we say that yes, this is the T stage. You can see the T stage. Perpendicular means it is a T stage. Okay. If it is at 90 degree, then we'll say that yes, it is the product has, the commodity has come to the harvesting stage. This T stage is majorly used in apple. Okay, based on the pedicel and the fruit surface angle, this T stage is majorly means we'll say like this is a harvestable maturity for apple fruits. Okay.
Okay,
So
sir.
like in exam, T stage is a major maturity indices for which commodity? Then it's apple.
And so what is the use of penetrometer?
Penetrometer. See, penetrometer, this is majorly used in peas in order to detect maturity indices of pea, like tenderness. See, in pea, pea is example for both for harvestable maturity and physiological maturity. See, for vegetable purpose, based on the consumer preference, we are harvesting in harvestable maturity, and for cereal purpose, we are harvesting in physiological maturity. So what this penetrometer do is, it is one of the texture analyzing device that is mainly used to see whether the product, whether the commodity pea is tender or not. Based on, actually, this penetrometer reading, it varies from for drying, it varies different readings. For freezing of the piece, it has a different reading, and for canning of the piece, it has a different reading. Means it is a device that is mainly used to detect whether the piece tender to eat or not.
Sir, penetrometer is different from tenderometer.
Means actually both are texture analyzing devices only. Means I no actually both are same. I think so. I'm not clear. I'll just clear. I'll ask seniors or my any other teachers, and I'll inform you. I think it's because both are different device, but the principle is same. In order to detect the freshness of the commodity or to detect the hardness of the commodity. Okay, like texture analyzer.
Okay, sir.
Okay.
Sir, what is the
Both
status of
principles
pectin?
same.
Sir,
Yes, yes.
what is
Yes, yes.
what is the status of pectin during ripening, sir?
Expectancy, I'll say this is very important question. See, before the commodity in as ripened, in the pre-ripened stage, what there will be there? There will be pectin, propectin will be there. Okay, in a commodity thing, for example, I'll take mango. Mango before the ripening, there will be propectin. Okay, this propectin is insoluble in water. This will be not soluble in water. As the mango starts ripening, when it comes from from immature stage to mature stage, the pectin content of the commodity starts decreases. Means from propectin, pectin will form. This pectin is water soluble. Okay, and after the mature, when it starts to over mature, there will be development of pectic acid by pectic methyl esterase enzyme. Okay, you just make a like a flow chart. First is propectin. Through help of propectinase enzyme, it will become pectin at the mature stage, and from pectin, it will convert to pectic acid at the over mature stage through pectin methyl esterase enzyme. Okay. In immature stage, the pectin will be more because the cell walls of the commodity there will be more calcium pectates or everything. So in mature it will be more. As a in mature stage also it will be more. As a over mature or a full at the end of stage, the pectin content decreases. Okay.
Okay, okay, sir.
Sir,
Okay.
ripening is directly related to ethylene evolution, no?
Yes, yes.
Then, sir, how it is inversely proportional in case of mango, in case of apple?
Apple, actually, I told Apple is Apple belongs to low section in respiration rate, and belongs to high section in ethylene evolution rate. Yes, ma'am. See, in Apple, based on study, what it is there is, see, for example, in Sapota, I'll say it will be more high. Yes, no. Because if you keep the Sapota in for 1 or 2 days, it starts ripening earlier. But if you keep apple, it takes more time. Means shelf life will be more. But for fully ripening, if you compare Sapota and apple, you can just analyze yourself. See, if you pluck the Sapota and keep in a place, within 3 or 4 days, it will start ripening. But apple, it doesn't ripe. So even though it exhibits a higher ethylene evolution rate, but the respiration rate is very lower. So that we can keep the apple for more days in cold storage, up to 6 months, up to 4 to 5, 6 months. Okay, it has higher ethylene but lower respiration rate is there. Okay.
Okay, sir.
Okay.
Sir, calendar dates means what, sir? According to Apple.
According to Apple, calendar dates means it's nothing but see, if you plant a tree, means you'll note it down that on this day I have planted this tree, and after 3 or 4 years after from first flowering, you will note down that yes, on this day, for example, 4th July, this is the day of flowering of the apple tree. Then after 125 or 125 days, you'll just think after this much days the apple should ripe. Means in calendar, you are thinking like after this number of days, the ripening or the maturity of the commodity starts. Means, so for example, in Sapota it is 300 days. In apple it is 125 days. Means from the date of flowering. For example, if it is if the apple flowers in July 4th, after 125 days or 130 days, you can analyze that yes, after this much days, the apple is going to ripe. Apple is going to mature. Okay, based on the number of days and months. Okay.
Sir, it is should to judge the harvest maturity of apple, sir.
What calendar? Yes, yes. In Apple, majorly, major maturity index is what and all is there. Days from full bloom to harvest, one, and another one is this one. The what is that? Starch index. That is also one major, and calendar dates is also one major, and also TSS also. These four are major in Apple, deciding the Apple, and also T stage. T stage. It is a major in Apple, deciding the maturity indices of Apple.
Sir, which instrument is used to measure the pectin contents?
Pectin content is measured by gel meter. Okay. Pectin, see, while we say regarding pectin, pectin it is measured by gel meter instrument or a device. It mainly works from 21 to 35 degrees Celsius temperature range. Okay. Pectin is measured used for jelly. You just this majorly from pectin they'll ask only this question. Gel meter. It is used for jelly making, during jelly making, and temperature range for jelly meter is 21 to 35 degrees Celsius. Okay, these two, these three.
So tendometer and penetrometer is same.
Actually, I also, when you asked, I also got some doubts. Okay, I'll just look it on it and I'll just inform. Okay, but the principle is same. But the principle is same to detect the tenderness of the particular commodity. I think device is different, but the principle is same. Okay, because I also have some confused. While you ask, I also started analyzing. I'll just see and I'll just inform. Okay, in next class tomorrow there is one more class. I'll just say that. Okay.
Yes.
Sir, during ripening, phenolic compound, what increase or decrease?
No, see, when ripening now, majorly the vitamins, minerals, and phenolic compounds. These three factors will be stable in ripening stage. But after when compared to immature and the mature stage, now the phenolic compounds will develop in a particular commodity, so that vitamins and minerals will be lesser in ripened fruits. Okay, vitamins and minerals will be more in immature fruits and it will be lesser in ripened fruits. But phenolic compounds, it will be more in ripened fruits. Okay, means the commodity starts developing the phenolic compounds like anthocyanins, quercetin, delphinidin, various pigments, various phenolic compounds. Okay, it will increase.
Okay, sir.
Sir, what is mean by T stage, sir?
Stage, I told just now, I think. Okay.
Actually, students, you are asking again and again same questions. Okay. If there is any other question except T stage, you can ask. Hanmesh, you can explain T stage once again. Otherwise, we will close the meeting.
Oh, okay, sir. Okay, sir. See, T stage, T stage mainly it is the angle between the pedicel of the apple. Majorly, T stage is used for detecting the maturity indices of apple. It is the angle between the pedicel of the fruit and the apple fruit, so that the pedicel will be straight in T stage. If it is a 90 degree, then we say that apple is a matured fruit. It is just the angle between pedicel and the apple fruit. Okay.
Okay, sir.
Hello. Yes.
Sir, which instrument is used to measure the oil content in avocado?
Oil content in avocado. See, oil content, these are majorly detected by Shock-Gillette method. There is one Shock-Gillette equipment where the petroleum ether solvent is used, and we are going to detect the oil content of the not only avocado, all the food commodity which are, we'll say like this commodity having this much oil content. No? These are majorly detected by Shock-Gillette method. Okay. Majorly petroleum, by using the petroleum ether solvent, we are going to use detect the oil content in avocado. Okay.
Okay, sir.
Okay.
So what is meant by the it, sir, in judging that harvesting humidity or any maturity of vegetables, how it is suitable to judge the maturity?
I told now like TSS, then in vegetables, when you come to vegetables, for example, in some for example, in like muskmelon and watermelon, TSS is major. Means it depends on individual commodity. Like for Tomato, if you see based on color, you will detect the maturity indices. Na? For canning, you are going to use overripe fruits. For export purpose, you are going to that turning stage or breaker stage, like that. Based on color, based on TSS, or in watermelon, based on yellowing at the lower surface in the when the fruit touches to ground, na? Yellowing of the lower surface or tapping, like that. It depends on the individual commodity to detect the maturity and indices. Okay.
Okay, sir.
Sir, during low astringency, what will be the tannin portion, sir? And it is soluble or
Oh,
insoluble?
tannin content, na?
Yes, sir.
When it is mature, tannin content. Actually, I also don't know. I'll just see and inform you. Okay. What was your question? At maturity, what is the tannin content, na?
During low astringency, what is the position of tannins? It is soluble or
I
insoluble?
see. I'll soluble. I don't know whether it is soluble or insoluble, but majorly astringency of the fruits is mainly due to tannin content, like in persimmon and other fruits. Na? I just know that tannin content is responsible for astringency, but I don't know whether it's soluble or insoluble. I'll just see it and I'll just inform you. Okay.
Okay, okay, sir.
Okay. Thank you.
Sir, what is been the harvesting index, sir?
Harvesting index.
Uh,
Harvesting index. It's majorly means it's we are going to detect the harvesting of the commodity. Like I told, na, like various maturity indices, various equipments you are going to use, various TSS or any other you are going to use, or various mechanical methods like Tapka stage or chemical methods like starch. These are all comes under single is harvesting index or maturity index of a particular commodity. Okay.
Actually, the harvest index is the index of the crop when the crop should be harvested. This is
Yes,
simple
yes.
answer.
Yes.
Okay. You go through the different crops and different harvesting indices. Those questions will be asked. They won't ask any concept questions. So please don't scratch your head in understanding the concept. Like what will happen during maturity, what are the compounds will be accumulated. Don't scratch your head in unnecessary things, because it's a kind totally energy wasting, time wasting. So only read what they will ask in the exam. Okay. So you go through the question papers, definitely you will find the questions how they will be asked. Okay.
Yes.